Hello, everybody. Welcome back to the Succinct Psychology, little intro to psychology course. My name is Prescott. Again, I'm here with Succinct Psychology, and today we are going to be starting Chapter 3, Biopsychology. Again, this is the OpenStax Psychology 2E textbook that we're going to be going through. And so this is a bit of a longer chapter, so it'll probably be one or two extra little videos. But without further ado, let's get started. So, first slide here, we have biopsychology. That's the unit. Biopsychology explores the biological mechanisms that underlie behavior. Among many things, it studies genetics, focusing on how inherited genes can affect not just the physiological, but psychological traits of a person. So physiological is physical aspects, where psychology is mind. The structure and function of the nervous system, and how the nervous system interacts with the endocrine system. So here we have a nice picture. So different brain imaging techniques provide scientists with insight into different aspects of how the human brain functions. So left to right, we have a PET scan, positron emission tomography, so right here, a CT scan, computed tomography, and an fMRI, functional magnetic resonance imaging. So these three are types of scans, obviously, and we'll go more into depth about each of these later in the unit, so I won't touch on it right now. All right, human genetics studying human genetics can help researchers understand the biological basis underlying the different behaviors, thoughts, and reactions of humans. So, why do two people infected by the same disease have different outcomes? Are there genetic components to psychological disorders such as depression? And how are genetic diseases passed through family lines? So these are kind of some questions you ask when talking about human genetics. Continuing on, we have the theory of evolution, which I'm sure you guys have heard about and read about in your high school science courses. So Charles Darwin explored the concept of inheritance of traits throughout generations in his theory of evolution through natural selection. So the organisms that are better suited for their environment will survive and reproduce, while those that are poorly suited for their environment will die off. So that's kind of his theory there. Characteristics and behaviors that impact survival and reproduction. Those that help protect against predators, those that increase access to food, and those that help offspring to live. So here's a little quote from Charles Darwin. "It is not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is most adaptable to change." So that's very obviously just saying that it doesn't matter how strong an organism is, it's the one that's able to adapt to changes in its environments. So that's the theory of evolution. Nice little refresher for you. So here we have Charles Darwin. So in 1859, Charles Darwin proposed his theory of evolution by natural selection in his book On the Origin of Species. So figure B here, the book contains just one illustration. This diagram shows how species evolve over time through natural selection. And so you can see it's a pretty complex little graph he's got here, but just kind of breaking down how species change and evolve over time. Continuing with the theory of evolution, we have a little question here. So why have certain genetic diseases that cause people to die not become less common? So for an example, we have sickle cell anemia. So sickle cell anemia is a genetic condition in which red blood cells take on a crescent-like shape affecting how they function. This causes many people to die at an early age, but it is still common among people of African descent. Carriers of only one copy of the sickle cell gene are thought to be immune from malaria, a deadly disease which is common in Africa. In this example, carrying the gene makes a person better suited for their environment. So this is kind of explaining, it's a genetic disease and it causes people to die early, but in their environment, it's actually beneficial to have one copy of the sickle cell gene. So that's why it hasn't been, phased out of, that population. So here we have a picture. So you can see normal blood cells, nice and round, and sickle-shaped blood cells. You can see they're shaped like sickles. Normal blood cells travel freely through the blood vessels, while sickle-shaped cells form blockages, preventing blood flow, which is obviously bad, can lead to heart attacks, blood clots, just things you don't generally want to have to deal with, if you want to live long at least. Genetic variation. So the genetic difference between individuals. So genetic variation contributes, sorry, I'm slipping on my words, to a species' adaptation to its environment. So it begins when an egg, containing 23 chromosomes, is fertilized by a sperm, also containing 23 chromosomes. Now, a chromosome is a long strand of genetic information known as DNA. Now, DNA is deoxyribonucleic acid, or DNA, is a helix-shaped molecule made of nucleotide base pairs. In each chromosome, sequences of DNA make up genes. And a gene is a sequence of DNA that controls or partially controls physical characteristics known as traits. So that'd be your eye color, your hair color, etc. A gene may have multiple possible variations or alleles, a specific version of a gene. So a specific gene may code for hair color, and the different alleles of that gene affect what the hair color will be. Here we have genotype versus phenotype. So figure A is a genotype refers to the genetic makeup of an individual based on the genetic material, DNA, inherited from one's parents. Now, a phenotype describes an individual's observable characteristics, such as hair color, skin color, height, and build. So genotypes are kind of like this cellular genetic material you can't physically see, whereas a phenotype is, well, what you can physically see. You can think of it as physical. So it's something that the naked eye can see. You can tell somebody's skin color and hair color and whatnot. Next, we have dominant versus recessive alleles. So the majority of inheritable traits are controlled by more than just one gene, and in such cases, they are known as polygenic traits. Some traits are controlled by one gene. Alleles can be dominant or recessive. In the example below, A is the dominant allele for flower color, purple, and A, so lowercase, is the recessive allele for flower color, white. So possession of a dominant allele will always result in expression of that phenotype. This could be inherited from one parent, so here you have dominant, recessive, capital A, lowercase a, or both parents, so two dominant alleles. The phenotype of a recessive allele will only be physically expressed if the person is homozygous for that allele, meaning they inherited a recessive allele from both parents. So here you have the two recessive alleles, lowercase a and lowercase a. So heterozygous is consisting of two different alleles, so dominant and recessive, and homozygous is consisting of two identical alleles, so dominant, both recessive. And so here we have a nice picture kind of breaking that down, making it more digestible. So A, the dominant allele, is for the color purple in the flower, while the recessive allele, lowercase a, is the allele for the white color. So here we have, if you have the two dominant alleles, you're going to get a purple flower. But if you have two of the recessive alleles, you'll get a white flower. But if you have a dominant and a recessive, it's going to be the dominant allele color, which is purple. So we're going to go more into this with Punnett Squares. This is also something you guys probably learned about at some point. So here we have two figures, A and B. B is a nice cleft chin. So a Punnett Square is a tool used to predict how genes will interact in the production of offspring. The capital B represents the dominant allele, and the lowercase b represents the recessive allele. In the example of the cleft chin, where B, so dominant, capital B, is cleft chin, whenever a pair, or wherever a pair contains the dominant allele B, you can expect a cleft chin phenotype. And again, phenotype is the physical things that you can see. So you can expect a smooth chin phenotype only when there are two copies of the recessive allele, so lowercase b, lowercase b. So in this figure, you can see the cleft chin is shown here, and it's an inherited trait. And so to break down this Punnett Square thing, in case it seems confusing, it's actually really simple. So you have two axes here, and here's the father's allele, so he has two recessive alleles, and then so he's homozygous recessive. And then you have the mother, who is heterozygous, and she has a dominant and a recessive. So all you have to do is, make these four little squares and put the two parents' alleles, on each side. So you have your lowercase b, uppercase B, lowercase b, and then all you do is drop this capital B down and then bring over the father's recessive. So in this circumstance, that would result in the dominant allele making itself present. And that's just all you really have to do. I won't get too much into it, but again, just drop, bring over. It's going to have the dominant allele, and then drop, bring over, recessive, recessive. So pretty straightforward. So continuing with Punnett Squares, In this Punnett square, N represents the dominant allele, and P represents the recessive allele that is associated with PKU. If two individuals mate who are both heterozygous, so you have the dominant and the recessive, for the allele associated with PKU, their offspring have a 25% chance of expressing the PKU phenotype. So where do harmful genes like PKU come from? Some are the result of mutations, and a mutation is a sudden permanent change in a gene. Many mutations are harmful, but some can also be beneficial. So, breaking it down more, here we have another Punnett square. Same thing, set it up. So you have the two heterozygous parents. In this Punnett square, N represents the normal allele, and P represents the recessive allele that is associated with PKU. If two individuals mate who are both heterozygous for the allele associated with PKU, their offspring have a 25% chance of expressing the PKU phenotype. So you can see here, just remember, drop, bring over, drop, bring over, drop, bring over. So there's only a 25% chance, because 25, 50, 75, 100, that the offspring will express the PKU phenotype. Continuing on. So we'll make this the last slide for this video, but we'll continue in the next. So anyways, gene-environment interactions. So nature and nurture work together like complex pieces of a human puzzle. The interaction of our environment and genes makes us the individuals we are. There are many different ways to look at this interaction. So we have range of reaction, which asserts our genes set the boundaries within which we can operate, and our environment interacts with the genes to determine where in that range we will fall. Then we have genetic environmental correlation, which is a view of gene-environment interaction that asserts our genes affect our environment, and our environment influences the expression of our genes. And then we have epigenetics, which is a study of gene-environment interactions, such as how the same genotype leads to different phenotypes. So we will stop there for today. Again, my name is Prescott, and I'm here with Succinct Psychology, and I hope to see you guys in the next video where we will continue Unit 3. All right, have a good one. Bye. Hello, everybody. Welcome back to Succinct Psychology, or Succinct. My name is Prescott, and we're going to continue Unit 3 Biopsychology from the OpenStax online textbook, Psychology 2E. And let's go ahead and keep it on. So last time, went over gene-environment interactions, Punnett squares, phenotypes, genotypes, theory of evolution. And so today, we're going to continue on, and we're going to talk a little bit about neuron structure and axon cells, all that fun stuff. So, continuing on from last time, we have neuron structure. So the function of a neuron is aided by specific aspects of its structure. Its semi-permeable membrane allows smaller molecules, or molecules without an electrical charge, to pass through it, while stopping larger or highly charged molecules. Incoming electrical signals from other neurons are received by the dendrites, which then send the signal down the axon. So here we have a figure. This illustration shows a prototypical neuron, which is being myelinated. So axons are covered in a myelin sheath made of a fatty substance that insulates axons and allows the signal to travel down the axon quicker. At the end of the axons are terminal buttons, which contain synaptic vessels, so storage sites for chemical messengers called neurotransmitters. So here we have the axon, you can see the myelin sheaths, and then these terminal buttons, which contain the synaptic vessels, and we will go into what the synapse is right here. So the synapse is the space between the terminal button of one neuron and the dendrite of another. And so you can see here the terminal buttons, these are the dendrites of another neuron. So the synapse is that space in between the two. In this pseudo-colored image from a scanning electron microscope, a terminal button, green, has been opened to reveal the synaptic vesicles, orange and blue, right here, inside. Each vesicle contains about 10,000 neurotransmitter molecules. So, pretty interesting. Now, at resting potential, Na positive, these blue pentagons, are more highly concentrated outside the cell in the extracellular fluid, shown in blue. whereas K positive, purple squares, shown here, are more highly concentrated near the membrane in the cytoplasm or intracellular fluid. So extra is outside the cell, intra is in. Other molecules, such as chloride ions, these yellow circles, seen here, and negatively charged proteins, big brown squares, help contribute to a positive net charge in the extracellular fluid and a negative net charge in the intracellular fluid. So it's positive, negative. So going on, we have action potential. So neurotransmitters from nearby neurons attach to receptors on dendrites, causing the membrane potential to change. So depolarization is when the membrane potential becomes less negative, making the neuron more likely to fire, which is called excitation, making it more likely to fire, exciting it. Hyperpolarization is when the membrane potential becomes more negative, making the neuron less likely to fire, which is inhibition. It's making it less likely to fire. Excite, more likely. Inhibit, less likely. If the level of charge reaches the threshold of excitation, an action potential will occur. Ion channels open, causing Na positive to rush into the cell and the inside to momentarily become more positive. So the threshold of excitation is a level of charge in the membrane that causes the neuron to become active. And action potential is an electrical signal. So action potentials act on an all or none principle. The incoming signal is either sufficient to reach the threshold of excitation or it is not. So there's no halfway reaching the threshold. It's all or nothing. Here we have a little graph. During the action potential, the electrical charge across the membrane changes dramatically. As you can see here, you start with your resting potential, reach the threshold of excitation, you reach your peak action potential, and then you start to repolarize and go into hyperpolarization, which is again making it less likely to fire. Now we have reuptake. So once an action potential has occurred, excess neurotransmitters in the synapse either drift away, are broken down, or are reabsorbed. So reuptake involves moving a neurotransmitter from the synapse back into the axon terminal from which it was released. So reuptake involves moving a neurotransmitter, from the synapse back into the axon. Oh, it just repeats itself, that's weird. So here you can see a little process of reuptake, neurotransmitter goes back into the synapse so it can be released again between the terminal buttons and dendrites. So what are neurotransmitters? So neurotransmitters is a chemical messenger of the nervous system. Different neurons release different types of neurotransmitters that have many different functions. So from a biological perspective, it's a view that psychological disorders like depression and schizophrenia are associated with imbalances in one or more neurotransmitter systems. So here are some neurotransmitters. We have acetylcholine, which dictates muscle action and memory, beta endorphin, which is pain and pleasure, dopamine, which is mood, sleep, and learning, norepinephrine, heart, intestines, and alertness, and finally serotonin, which is mood and sleep. So there are some drugs for neurotransmitters. So first we have psychotropic medication, so drugs that treat psychiatric symptoms by restoring neurotransmitter balance. So an agonist is a drug that mimics or strengthens the effect of a neurotransmitter, while an antagonist is a drug that blocks or impedes the normal activity of a given neurotransmitter. So easy way to remember that, antagonist of a story is trying to block or impede the hero, so do antagonist drugs. But they're doing it with neurotransmitters, not heroes of the story. So agonist/antagonist drugs are prescribed to correct neurotransmitter imbalances. Example, Parkinson's disease is associated with low levels of dopamine. Dopamine agonists are often prescribed as one form of treatment. So again, dopamine is mood, sleep, and learning, so it's trying to strengthen the effect of the dopamine. While schizophrenia, on the other hand, is associated with too much dopamine. So many antipsychotic drugs are therefore dopamine antagonists. They're trying to impede the normal activity of that dopamine neurotransmitter because there's too much of it. So going on, we have the parts of the nervous system. So the nervous system is divided into two major parts. The central nervous system, shown here is the brain and spinal cord, and the peripheral nervous system, which is all the different nerves. So the peripheral nervous system is made of two different parts. The somatic nervous system, which relays sensory and motor information to and from the central nervous system, and the autonomic nervous system, which controls our internal organs and glands and can be divided into the sympathetic and parasympathetic nervous systems. So the sympathetic nervous system is involved in stress-related activities and functions, and it prepares us for fight or flight. Now, I'm sure you've heard of the fight or flight response, but just to give you a definition, is an activation of the sympathetic division of the autonomic nervous system, allowing access to energy reserves and heightened sensory capacity so that we may fight off a given threat or run away to safety. So, the sympathetic nervous system, it triggers your fight or flight response in case, you're in a dangerous situation and you either need to be able to defend yourself or get out of there. Now, the parasympathetic nervous system is associated with routine, day-to-day operations of the body under relaxed conditions, and also has the rest and restore response, which relaxes the body after fight or flight, aka rest and digest. So sympathetic nervous system gets you ready for fight or flight, and parasympathetic calms you down after you're out of that situation. So the sympathetic and parasympathetic nervous systems complement each other to maintain homeostasis, which is a state of equilibrium in the body. Here we have subdivisions of the nervous system. So starting off, you have the nervous system, which can be divided into the central nervous system, brain and spinal cord, and the peripheral, which is nerves to and from your central nervous system. Now, the peripheral can be broken down more into the somatic and autonomic nervous systems. So somatic is touch, hearing, etc., and muscles that move the body. And then autonomic are the internal senses for gut, smooth muscles and gut, etc. And autonomic has two more branches, which is the sympathetic, emergency, and parasympathetic, which is housekeeping. So just relaxed state, day-to-day stuff. So we're going to stop this video here. We will continue to go into the nervous system, brain and spinal cord, all that fun stuff in the next video. Again, my name is Prescott here at Succinct Psychology, and I hope that you guys tune in for the next video. Have a great day, and see you next time. Welcome back, everybody. My name is Prescott. I'm here at Succinct Psychology, and we are going to continue going through Unit 3 Biopsychology from the OpenStax textbook. Now, last time we talked about the nervous system, the central nervous system, mainly the peripheral nervous system, different parts of that, the somatic nervous system, autonomic nervous system, your fight or flight response, your calm down response, all that fun stuff. And today we're going to continue with this nice little graph of the autonomic nervous systems. So here you have parasympathetic and sympathetic. Now, the sympathetic and parasympathetic divisions of the autonomic nervous system have the opposite effects on various symptoms. So you can see from this little graph, the parasympathetic, it constricts your pupils, stimulates salivation, slows heart rate, constricts bronchi, stimulates digestion, and causes bladder to contract, whereas the sympathetic does the exact opposite of each of those. So it dilates your pupil instead of constricts it. It inhibits your salivation instead of stimulates it, and so on and so forth. Now, going on, we're going to get into the brain and spinal cord. So the brain is comprised of billions of interconnected neurons and glia. It is bilateral, which means it's two-sided, and can be separated into distinct lobes, but all areas interact with one another. Now, the spinal cord delivers messages to and from the brain. It has its own system of reflexes, and the top merges with the brainstem, and the bottom ends just below the ribs. So it's functionally organized into 30 segments, each connected to a specific part of the body through the PNS, peripheral nervous system. Sensory nerves bring messages in and up to the brain. Motor nerves send messages out to the muscles and organs. In moments of survival, automatic reflexes allow motor commands to be initiated without sending signals from sensory nerves to the brain first, allowing for very quick reactions. So that would be when you're in the fight or flight mode, your automatic reflexes, you can initiate your motor commands without having to set your nerves having to send signals to the brain first. It can be just automatic flip of a switch reactions in survival situations. So going more into the brain, we have the two hemispheres. Now, lateralization is a concept that each hemisphere of the brain is associated with specialized functions. so that your left hemisphere controls the right side of your body, and your right hemisphere controls the left side of your body. So the surface of the brain is covered with gyri and sulci, and the sulci, well, a deep sulcus is called a fissure, such as the longitudinal fissure that divides the brain into left and right hemisphere. So here you can kind of see, but we'll see it better in a second, the longitudinal fissure that separates the two hemispheres of the brain, and then you have your sulci and your gyri. So the corpus callosum. So here we have some pictures. So the corpus callosum connects the left and right hemispheres of the brain. So you can see corpus callosum here. Now, a scientist in this picture spreads this dissected sheep brain apart to show the corpus callosum between the hemispheres. So you can see it very clearly right there, a little bit of a gross picture, but it helps you to better visualize what the corpus callosum is, where it's at. So now we're going to go more in depth into the different parts of the brain. So we have the forebrain, midbrain, and hindbrain. So the brain's parts can be divided into these three main categories. Again, it's the forebrain, midbrain, hindbrain. Now, going into the forebrain, we have forebrain structures. So the forebrain is the largest part of the brain. It contains the cerebral cortex, which is for higher level processes, the thalamus, sensory relay, hypothalamus, homeostasis, pituitary gland, master gland of the endocrine system, and the limbic system, which is a motion and memory circuit. And we'll go more into these here. So, cerebral cortex is the surface of the brain that's associated with our highest mental capabilities, such as consciousness, thought, emotion, reasoning, language, and memory. It can be broken up into 4 lobes, each with a different function. So, frontal lobe, temporal lobe, occipital lobe, and parietal lobe. Now, I can't give you the best way to memorize in case you need to where each lobe is. I just always was able to remember it as, frontal and occipital is in the back. And then, well, you can think of temporal, your temple, so around this area, and then parietal, just remember it's kind of up here on the top. So the frontal lobe is involved in executive functioning, planning, organization, judgment, attention, and reasoning, motor control, emotion, and language. It contains the motor cortex, which is a strip of cortex involved in planning and coordinating movement, the prefrontal cortex, which was responsible for higher level cognitive functioning, and Broca's area, region in the left hemisphere that is essential for language production. So damage to Broca's area leads to difficulties producing language. So here we have a classic example, damage to the frontal lobe. Phineas Gage, he's very famous because of this little story here. So while working as a railroad foreman, an accident caused an iron rod to penetrate through Gage's skull and frontal lobe. After the accident, people noticed changes in his personality. So before the accident, he was well-mannered and soft-spoken, and after the accident, he started behaving in odd and inappropriate ways. So these changes were consistent with loss of impulse control, a function of the frontal lobe. So see here, so Phineas Gage holds the iron rod that penetrated his skull in an 1848 railroad construction accident. So, and then here's a picture of what the actual accident was like, and I think it's a miracle he survived. So Gage's prefrontal cortex was severely damaged in the left hemisphere. The rod entered Gage's face on the left side, passed behind his eye, and exited through the top of his skull before landing about 80 feet away. That's just crazy. But obviously having damage to your frontal lobe is not a good thing. Just having any kind of brain damage tends to not be very fun. So, continuing on, we have the parietal lobe. It is involved in processing various sensory and perceptual information. It contains the primary somatosensory cortex. Now the somatosensory cortex is essential for processing sensory information from across the body, such as touch, temperature, and pain, and it is organized topographically. So here we have a nice little visual of the cerebral cortex and just going through all the different parts of your body. So we're going to wrap up this little part here. The temporal lobe, so it is associated with hearing, memory, emotion, and some aspects of language. It is located on the on the side of the head near the temples, so again, temporal. And it contains the auditory cortex, which is a strip of cortex in the temporal lobe that is responsible for processing auditory information. And then it also has Wernicke's area, which is important for speech comprehension. Damage to Wernicke's area results in difficulty understanding language. So here we have Broca's area, and remember that's producing language, and then Wernicke's area, which is understanding. So damage to either Broca's area or Wernicke's area can result in language deficits. The type of deficits are very different, however, depending on which area is affected. So if you have damage to Broca's area and not Wernicke's area, the kind of language deficit you have is going to be very different. And so here we have just a slightly bigger picture saying the same thing. And so before we go more into different parts of the brain, I'm going to wrap up this video here so it's not too long, it's more digestible for everybody. So I hope to see you guys again. We're going to continue going through the different parts of the brain, different lobes, all that fun stuff. Again, my name is Prescott here at Succinct Psychology, and I hope to see you guys in the next video. Have a good one. Bye-bye. Hello everybody, welcome back. This is the final video of Unit 3, Biopsychology from the OpenStax textbook. My name is Prescott, I'm here with Succinct Psychology, and we're going to continue from where we left off. Last time we were just going through all different parts of the brain, lobes, what they do, all that cool stuff, and so we're going to continue going through that because there's a lot of parts in the brain, so it's very in-depth here. So, starting off, we have the occipital lobe, which is associated with visual processing. It contains the primary visual cortex, which is responsible for interpreting incoming visual information, and is organized retinotopically. So here, occipital lobe, remember, back of your head, kind of. So, starting next, we have the thalamus. So the thalamus serves as the relay center of the brain where most senses, excluding smell, are routed before being directed to other areas of the brain for processing. So the thalamus serves as the relay center for the brain, or most center for processing. Next, we have the limbic system. So the limbic system is involved in mediating emotional response and memory. It is made up of a number of different structures, some of the most important ones being the amygdala, which is involved in our experience of emotion and tying emotional meaning to our memories, involved in processing fear, the hippocampus, which is the structure associated with learning and memory, in particular spatial memory, and then the hypothalamus, which regulates homeostatic processes, including body temperature, appetite, and blood pressure. So you can see where each of those are, and the way I remember these is amygdala. There's a game I played before where there's this big, scary spider monster thing called an amygdala. And so I'm like, oh, amygdala is a big, scary monster. Amygdala is involved in processing fear. And I have that memory, that emotional memory of being like, oh, that's kind of spooky. So that's your amygdala. Hippocampus, which is learning and memory. And you can think of it as if you ever saw hippos on campus, you would remember that. And then hypothalamus, which one of my professors told me is like hypo the llamas or something, because processes body temperature and your appetite and blood pressure, so you're hypoing the llamas, whatever that means. So those are the limbic system. And again, we have another nice picture of the limbic system. Next, we have the midbrain. That's weird, I don't know why that's saying. So the midbrain is made up of the reticular formation, which is important in regulating the sleep-wake cycle, arousal, alertness, and motor activity. The substantia nigra, where dopamine is produced, involved in control of movement, and the ventral tegmental area, VTA, where dopamine is produced. Again, that's learning, memory, mood, and is associated with mood, reward, and addiction. So degeneration of the substantia nigra, and VTA is involved in Parkinson's disease. So we have the substantia and the VTA. So degeneration of that is what leads to Parkinson's, and Parkinson's is not a fun thing to have. So here again, you can see where they're located in the midbrain. Next, we have the hindbrain. So the hindbrain is made of the medulla, which controls automated processes like breathing, blood pressure, and heart rate. The pons, which connects the brain and the spinal cord, is involved in regulating brain activity during sleep. The cerebellum which controls our balance, coordination, movement, and motor skills, and it is thought to be important in the processing of some types of memory. So these three structures combined are known as the brainstem. So here you can see, again on the picture, you have the pons, medulla, and cerebellum. So this is what your brainstem is made up of. Once again, more zoomed in pictures, you can see a little bit better. So that's the hindbrain. So going on, we're going to go into brain imaging. So first off, we have computerized tomography, or CT scan. So you can just say CT scan, you don't have to say computerized tomography. That's a lot to say. It involves X-rays and creates an image through X-rays passing through varied densities within the brain. So a CT scan can be used to show brain tumors. So here we have some figures. So a CT scan can be used to show brain tumors. So figure A is the CT scan. The image on the left shows a healthy brain, whereas the image on the right indicates a brain tumor in the left frontal lobe. So you can see here, this is a nice healthy brain, but you can see this abnormality right here. That's a tumor. So CT scans are good for finding those and, helping you fix them, basically, knowing there's something to be fixed. So next we have positron emission tomography scans, or PET. So a PET scan is helpful for showing activity in different parts of the brain. So you can see here, it involves injecting individuals with a mildly radioactive substance and monitoring changes in blood flow to different regions in the brain. A PET scan is helpful for showing activity in different parts of the brain. So here you can see what a PET scan looks like. Next, we have MRI and an fMRI. So an MRI is magnetic resonance imaging. So magnetic fields are used to produce a picture of the tissue being imaged. And a functional magnetic resonance imaging, or fMRI, is an MRI that shows changes in metabolic activity over time. So MRI is just like a picture of the image, whereas, my mouse is acting over here. So just a straight picture of the image, while an fMRI is showing activity over time. So it's not just one image. So this image represents a single frame from an fMRI. So you can see it shows activity in the brain, but this is just one frame. Normally it shows over time how the brain is activated. Next we have an electroencephalography. I think I said that right. Or EEG to make it a bit easier. So it involves recording the electrical activity of the brain via electrodes on the scalp. Using cats with electrodes, modern EEG research can study the precise timing of overall brain activities by tracking amplitude and frequency of brain waves. So here you see a picture, electrodes on the brain, well, not on the brain, but on the head, so they can track, amplitude and frequency of your brain waves. And he's completing a task so they can measure all that good stuff. So the endocrine system is a series of glands that produce hormones to regulate normal bodily functions. The hypothalamus links the nervous system and endocrine system by controlling the pituitary gland. So pituitary gland serves as the master gland, controlling the secretions of all other glands. The thyroid secretes thyroxine, which regulates growth, metabolism, and appetite. Adrenal gland secretes hormones involved in the stress response. Gonad, they secrete sex hormones, which are important for successful reproduction and regulate sexual motivation and behavior. And then you have pancreas, which secretes hormones that regulate blood sugar. So here you have a nice little image showing where these different parts of your endocrine system are. You have pineal gland up here, it's like right here in your brain, thalamus, pituitary gland. You have the thymus, the thyroids, adrenal glands. You can kind of see for yourself. You don't need me to break down everything for you. So yeah, major glands of the endocrine system are shown here. And finally, we're going to just have a little reminder of the major neurotransmitters and how they affect behavior. So going down the list, we have acetylcholine, which is involved in muscle, action, and memory. And potential effect on behavior is it can lead to increased arousal and enhanced cognition. Beta endorphin, which is involved in pain and pleasure, and it can have an effect on behavior by decreasing anxiety, and decreasing tension. Dopamine, mood, sleep, and learning, and potential effect is increased pleasure and suppressed appetite. Then gamma aminobutyric, aminobutyric acid, or GABA, which is brain function and sleep, and is decreased anxiety and decreased tension. Then you have glutamate, which is memory and learning, and it can affect your behavior by giving increased learning and enhanced memory. Then norepinephrine, which is your heart, intestines, and alertness, and it can affect your arousal and suppress your appetite. And finally, serotonin, which is mood and sleep, and the potential effect is a modulated mood and a suppressed appetite. So that's a nice list of neurotransmitters. If you want to study, you can come to this slide, just a quick reminder of the different neurotransmitters and what they're involved in and their potential effect on behavior. So, that was a much longer unit than I think the other ones we've done so far, but I hope you guys found it interesting. All this, different lobes and parts and neurotransmitters in the brain can be a little much to remember right off the bat, but hopefully, little insights I gave will be helpful in remembering all this stuff. And can always just keep coming back and watching these videos. And that is all for Unit 3, Biopsychology. We will continue Unit 4 next time, or not continue, but start Unit 4 next time. Again, my name is Prescott. I'm here at Succinct Psychology. This is the OpenStax textbook, and I hope you guys enjoyed these videos, and I will see you next time. Thank you. Bye-bye. Hello, everybody. My name is Prescott, and welcome back to Succinct Psychology. Today, we're going to continue going through the Psychology 2E OpenStax online PowerPoints and textbook. And today, we're going to be going over Chapter 4, States of Consciousness. So, without further ado, let's get going. So, starting off, what is consciousness? So consciousness is the awareness of internal and external stimuli, such as feelings of hunger and pain, or detection of light. Now, wakefulness is high levels of sensory awareness, thought, and behavior. So sleep, which we all experience, is a quiet and mysterious pause in our daily lives. So here, figure 4.1, you have two sleeping children depicted in this little 1895 painting. Just them sleeping, nothing much behind that. So let's continue on. So first off, we have biological rhythms. So a biological rhythm is an internal cycle of biological activity, including fluctuation of body temperature, an individual's menstrual cycle, and levels of alertness. Now, circadian rhythm is a biological rhythm that occurs over approximately 24 hours. So it is generated by the suprachiasmatic nucleus, or the SCN, or suprachiasmatic, my bad. Now, the sleep-wake cycle, one of our main circadian rhythms, is linked to our environment's natural light-dark cycle. So what controls our biological rhythms? So the hypothalamus, if you remember that from, I think it was last unit, maybe it was unit 2, but the hypothalamus is responsible for maintaining homeostasis, the tendency to maintain a balance or optimal level within a biological system. So going on. So here we got a nice chart. This chart illustrates the circadian change in body temperature over 28 hours in a group of 8 young men. Body temperature rises throughout the waking day, peaking in the afternoon, and falls during sleep, with the lowest point occurring during the very early morning hours. So, here you can see, start to wake up, body temperature rises, it peaks at around 9-10 p.m. And then once, that's when most people go to sleep, or at least the men in the study. And then body temperature pretty sharply drops down to a trough or low at 4 a.m. before the cycle begins again. So, the suprachiasmatic nucleus, almost made the same little error there, or the SCN. So, the suprachiasmatic nucleus, located in the hypothalamus, serves as the brain's clock mechanism. The clock sets itself with light information received through projections from the retina, allowing it to synchronize with the outside world. So here you see a little chart. Light goes in to the hypothalamus, and which also contains your suprachiasmatic nucleus. So nice little chart there. So continuing on, we have melatonin and sleep regulation. So our sleep-wake cycle is also regulated by other factors such as the hormone melatonin. Melatonin release is stimulated by darkness, making us sleepy, and inhibited by daylight. Melatonin is released in the pineal gland. So you see right here, that's where melatonin is released. So sleep regulation is the brain's control of switching between sleep and wakefulness, as well as coordinating this cycle with the outside world. Each person has an individual circadian pattern of activity known as a person's chronotype. And you can remember that as chrono, time. I think that's the Greek prefix for it or definition. But anyways, I could be wrong. So disruptions of normal sleep. Certain circumstances can throw off our circadian rhythms. For example, jet lag, symptoms resulting from the mismatch between our internal circadian cycles and our environment. So this can lead to fatigue, sluggishness, irritability, and insomnia. And so if you've ever, traveled on a plane to a different time zone, you definitely know what jet lag is because going from one place to another, and the place you're going to is 1 hour behind where you left from. Your body's thinking, so say you got into the next airport at 7 o'clock, their local time, but it's 8 o'clock from where you left. That would cause jet lag because it messes up your internal circadian cycle. So next we have rotating shift work, a work schedule that changes from early to late on a daily/weekly basis. It becomes difficult for a normal circadian rhythm to be maintained, and it can result in persistent feelings of exhaustion and agitation, sleeping problems, and can lead to signs of depression and anxiety. So bright light can be used to realign our biological clocks with the external environment. So here we have devices like this are designed to provide exposure to bright light to help people maintain a regular circadian cycle. They can be helpful for people working night shifts or for people affected by seasonal variations in light. So it's really important to keep your circadian rhythm, your internal clock in line, normal because, you don't want to be irritable or have insomnia. That's no fun. Next, we have sleep deprivation. So sleep debt is a result of insufficient sleep on a chronic basis, meaning it happens consistently and over time. Now, sleep rebound is when a sleep-deprived individual will tend to take a shorter time to fall asleep during subsequent opportunities for sleep. So that's, if you've been going through a period of insomnia or restless, you can't get a good night's rest, then once your sleep rebound is, if it normally takes you 30 minutes to go to sleep, like you lay in bed, don't touch your phone or anything, you're just trying to go to sleep, it might take you 15 minutes because your body recognizes that you need more sleep, and in that opportunity, it's easier for you to fall asleep to catch back up. So this figure illustrates some of the negative consequences of sleep deprivation. While cognitive deficits may be the most obvious, many body systems are negatively impacted by lack of sleep. So you can see in the brain, this is a little chart showing the negative consequences again. So you can cause irritability, cognitive impairment, memory lapses or loss, impaired moral judgment, severe yawning, hallucinations, and symptoms similar to ADHD. It can also impair your immune system, cause you to have a higher risk of diabetes type 2. It could cause an increased heart rate variability or risk of heart disease. And your muscles, increased reaction time, decreased accuracy, tremors, aches, and then, some other effects of sleep deprivation are growth suppression. So, if you're young and growing, you might not grow as tall as you should. Risk of obesity, and a decreased overall body temperature. So I'm not going to go through one by one of this chart. If you want to, you can pause the video and go through it for yourself, save us some time. But so we have it's chart here, sleep needs at different ages. So, 0 to 3 months, newborn 3 months. Recommended is 14 to 17 hours. It may be appropriate, so it's okay to have 11 to 13 or 18 to 19. However, it is not recommended that newborn gets fewer than 11 hours or more than 19 hours. And as you can see, just quickly glancing through this, the older we get, the less hours of sleep we really need, because in these stages, you're growing into your full size, so you need enough rest and sleep. So again, you don't have that growth suppression that can come with sleep deprivation. So that's that nice little thing. But anyways, so what is sleep? Now, sleep is a state marked by relatively low physical activity and a reduced sense of awareness. Sleep-wake cycles seem to be controlled by multiple brain areas, including the thalamus and hypothalamus, slow-wave sleep, and the pons, REM sleep, rapid eye movement. Now, sleep is associated with the secretion and regulation of many hormones, including, again, melatonin, follicle-stimulating hormone, luteinizing hormone, and growth hormone. So there should be a figure here. Never mind. The pineal and pituitary glands secrete a number of hormones during sleep. So here you have pituitary gland and pineal glands. So those are secreting those hormones that are going on while you sleep. Now, why do we sleep? So first off, we have sleeping as an adaptive function or an evolutionary hypothesis. So sleep is essential to restore resources that are expended during the day. And sleep is an adaptive response to predatory risk, which increase in darkness. But there's little evidence to support these explanations. Next, we have the cognitive function. So it focuses on sleep's importance for cognitive function and memory formation. And research shows that sleep deprivation results in disruptions in cognition and memory deficits. These impairments become more severe as the amount of sleep deprivation increases. Slow-wave sleep appears to be essential for effective memory formation. Now, some benefits of sleep: maintaining a healthy weight, lowering stress levels, improving mood, increased motor function, as well as many benefits related to cognition and memory formation. Now, I'm going to stop this video here. We will be going to brain waves during sleep and what those look like in the next video. So I hope you guys, stay tuned and watch that. Again, my name is Prescott. I'm here at Succinct Psychology, and this is the OpenStax 2E textbook. And I hope to see you guys in the next video. Thank you. Have a good one. Hello everybody, welcome back to Unit 4 of the OpenStax 2E Psychology PowerPoint/textbook. My name is Prescott. I'm here at Succinct Psychology, and today we're going to be continuing from where we left off about basically sleep, the different effects that sleep has on us, what sleep deprivation has on us. That's kind of what we went through last video. And in this video, we're going to be focusing on brain waves during sleep, different brain waves, and all about that. So without further ado, let's get started. So, like I said, brain waves during sleep. Brainwave activity changes dramatically across the different stages of sleep. Changes in brainwave activity can be visualized using EEG. And again, that's when they get electrodes, put it on your scalp, and they measure the electric activity of your brain. So first we have alpha brainwaves, which are relatively low frequency, relatively high amplitude, and are synchronized. So you see here we have alpha. And then we have theta, which is low frequency, low amplitude, and delta, which is low frequency, high amplitude, but they're desynchronized. So again, alpha, theta, you can see the differences in their amplitude and frequency, and then delta. You can see delta gets a little crazy with that. They're not super close together, the brain waves, that's frequency, but they have high amplitude. You can see how high up the different peaks go. So going more into depth with that, we have stages 1 and 2. So stage 1 is a transitional phase occurring between wakefulness and sleep. So rates of respiration and heartbeat slow down, your overall muscle tension and core body temperature decrease, and they contain alpha waves. Stage 1 has alpha waves. Now stage 2, the body goes into deep relaxation and starts producing beta waves, and is characterized by the appearance of both sleep spindles and K complexes. So, sleep spindle is a rapid burst of high-frequency brain waves, and K complexes are very high amplitude, you can see how high it goes, pattern of brain activity. So, again, frequency is how close these peaks and troughs are, and amplitude is how high up they go. So, stages 3 and 4, known as slow wave sleep, and we touched on that in the last video. And that's when you start to produce delta waves, and respiration and heart rate slow down even further. So, figure A, you can see these are delta waves, and then figure B, you see the slow wave stage 3 and 4 sleep. So, there's that. And next, we have rapid eye movement, or REM. So, as its name implies, rapid eye movements, paralysis of voluntary muscles, so you don't, having a dream, you don't start throwing punches or running around, running out of bed. And again, that's when you have dreams in REM and not in the other stages. And brainwaves are similar to those seen during wakefulness. So here, figure A, a period of rapid eye movement is marked by the short red line segment, which looks very similar to the brainwave seen in figure B during wakefulness. So you can see here's the REM brainwaves, and then when you're awake, here are the brainwaves there. So you can see how, if you look closely, you can see how similar the two are to each other compared to stage 1, stage 2, stage 3, and 4 brainwaves look very dissimilar to the awake ones. So next we have hypnogram of sleep stages. So a hypnogram is a diagram of the stages of sleep as they occur during a period of sleep. This hypnogram illustrates how an individual moves through the various stages of sleep. So you can see the hours of sleep they get, and then the corresponding stages with that. So it's very interesting. I actually didn't know about hypnograms, so I just learned something new. Now dreams. There are many theories on the meaning of dreams that vary across different cultures and periods of time. So Sigmund Freud, a member back in Unit 1, we briefly touched on his psychoanalysis of dreams, so it's a little throwback to that, but he saw dreams as a way to gain access to the unconscious, again, psychoanalysis. So two of his ideas were manifest content, or the actual content of the dream, and then the latent content, or the hidden meaning of the dream. So manifest content would be, you're at a park playing with a dog, that's what's actually happening in the dream, but then the latent content would be like, oh, what's the hidden meaning of that? What's your unconscious trying to tell you? Now, Carl Jung believed that dreams allowed us to tap into the collective unconsciousness. So collective unconsciousness is a theoretical repository of information shared by all people across cultures. And he believed that certain symbols in dreams reflected universal archetypes, which is really interesting idea that we have this kind of unconscious hive mind. So, or not hive mind, but just, what it says, that certain symbols are universal archetypes, even across cultures. Next, we have research. Research about dreams is dreams may represent life events that are important to the dreamer. Dreaming may represent a state of proto-consciousness or a virtual reality in the mind that helps a person during consciousness. So lucid dreams, certain aspects of wakefulness are maintained during a dreaming state. A person becomes aware that they are dreaming. And I've actually had lucid dreams before where I've been like, oh, I'm dreaming, and been able to like change things in my dream, or I would have a bad dream that was really unpleasant, and then at a certain point be like, oh, this is just a dream, and was able to force myself awake, because I was like, I don't want to be having this dream anymore. It's scary or upsetting. So lucid dreams are crazy. Now, insomnia is defined by difficulty falling or staying asleep for at least 3 nights a week for at least 1 month's time. So it is the most common sleep disorder and may be associated with symptoms of depression. Contributing factors include age, drug use, exercise, mental status, and your bedtime routines. Now, treatment for insomnia includes stress management techniques, changes in problematic behaviors that can contribute to insomnia, and cognitive behavioral therapy, or CBT, which focuses on cognitive processes and problem behaviors. So that's insomnia. Next, we have parasomnia. So parasomnias involve unwanted motor behavior / experiences throughout the sleep cycle. They include sleepwalking, which usually occurs during slow wave sleep. REM sleep behavior disorder occurs when the muscle paralysis associated with REM sleep does not occur. It includes high levels of physical activity during REM sleep and is often treated with clonazepam, an anti-anxiety medication. And earlier, I've mentioned how REM is important, that those muscles get shut down because, again, you don't want to be having high levels of physical activity. You don't want to be, hurting yourself in your sleep because you're having a dream where you're doing something very energetic. That's not good. Next, we have Restless Leg Syndrome, which involves uncomfortable sensations in the legs when trying to fall asleep that are relieved by moving the legs. It can be treated with a variety of medications. And then night terrors. Those are never fun. Sleeper experiences a sense of panic and may scream or attempt to escape, and they occur during and REM sleep. So there's parasomnia for you. So now you know that sleepwalking is parasomnia. I'm sure everybody knows about sleepwalking, so you know a little bit more about it. All right, next we have sleep apnea. So it occurs when individuals stop breathing during their sleep, usually for 10 to 20 seconds or longer. So it causes repeated disruptions in sleep and lead to increased levels of fatigue. Common in people that are overweight, and there are two types. Obstructive is when the airway becomes blocked and air is prevented from entering the lungs, and central is when the CNS fails to initiate breaths. So treatment can include a continuous positive airway pressure, or CPAP device, which pumps air into a person's airways. You can see right here, figure A and B, those are some CPAP devices, so that can help sleep apnea. And then we have sudden infant death syndrome, SIDS, which is as bad as the name implies. So it occurs when an infant stops breathing during sleep and dies. Infants younger than 12 months are at the highest risk, and boys have a greater risk than girls. So contributing factors include premature death, smoking within the home, and hypothermia. So here we have Safe to Sleep campaign, which educates the public about how to minimize risk factors associated with SIDS, so infant death syndrome. This campaign is sponsored in part by the National Institute of Child Health and Human Development. And I think we're going to wrap up this video here. We got over insomnia, sleep stages, rapid eye movement, sudden infant death syndrome, sleep disorders. So we will continue in the next video, starting off with narcolepsy, another sleep disorder, and just continuing on from there. Again, my name is Prescott. I'm here at PsychSync, Succinct Psychology, and this is the OpenStack Psychology 2E textbook, and I hope to see you guys in the next video. Have a great one. Bye. Hello, everybody. Welcome back to PsychSync, Succinct Psychology. Again, my name is Prescott, and we're going to finish up Unit 4, States of Consciousness, today. And sorry, I had a little brain fart there. But let's continue on from where we left off last time. Last time we started up sleep disorders, the effects of those, and also sleep stages. And so we're going to finish up the sleep disorders with narcolepsy. So narcolepsy involves an irresistible urge to fall asleep during waking hours. It is often triggered by states of heightened arousal or stress. It shares many features of REM, rapid eye movement sleep, including cataplexy, which is a loss of muscle tone while awake, or in some cases, complete paralysis of the voluntary muscles, and hypnagogic hallucinations, which are vivid, dreamlike hallucinations. So treatment for narcolepsy is psychomotor stimulant drugs. And if you guys want to have a little laugh, there's a video one of my professors showed us when we were learning about narcolepsy of a narcoleptic dog that you can find on YouTube, and it's like kind of sad but kind of funny because it would get too excited and just collapse and fall asleep. So if you guys want to check that out, I think that's pretty humorous, and you learn a bit more about narcolepsy there. So going on from sleep disorders, we have substance use disorders. So substance use disorder is a compulsive pattern of drug use despite negative consequences. That's the DSM-5 edition. It involves physical and psychological dependence on the drug. And so you have the physiological dependence, physical. It involves changes in normal bodily functions and withdrawal upon cessation or ceasing, stopping of use. And a psychological dependence is an emotional need for the drug. Then continuing with disorders, we have tolerance, which occurs when a person requires more and more of the drug to achieve effects previously experienced at lower doses, and it is linked to a physical dependence. And then withdrawal are negative symptoms experienced when drug use is discontinued. So not good stuff. So here we have a nice diagram of drug categories. So you have stimulants, depressants, and hallucinogens, and then you have all these different drugs in here. And it goes a little bit more separate circles. So psychomotor stimulants like amphetamines, cat, Ritalin, cocaine. You have sedative hypnotics, alcohol barbiturates, ether, GHB, and then you have certain drugs that are combinations of multiple categories. So like marijuana is a combination of a depressant, a stimulant, and a hallucinogen. Continuing on with the drug categories, and I won't go through every example and every effect used with each class of drug because I don't want this video to be too long, but starting off, and you can see right away that they're all psychologically addicting. So we have stimulants, so cocaine, amphetamines, MDMA, ecstasy or Molly. Effects on the body include increased heart rate, blood pressure, and body temperature. And then you can see it has a whole lot of effects when used, like increased alertness, mild euphoria, decreased appetite in low doses, and high doses increase agitation, paranoia, and can cause hallucinations. I was about to say hallucinogens. Next class, we have sedative hypnotics or depressants, like alcohol, barbiturates, benzodiazepines, and these can cause or will cause decreased heart rate and blood pressure. Low doses increase relaxation, decrease inhibitions. High doses can induce sleep, cause motor disturbance, memory loss, decreased respiratory function, and death in extreme cases. Next, you have opiates such as opium, heroin, fentanyl, morphine, and these cause decreased pain, pupil dilation, decreased gut motility, decreased respiratory function, and effects when used are pain relief, euphoria, sleepiness. High doses can cause death due to respiratory depression. And finally, we have hallucinogens. So marijuana, LSD, peyote, mescaline, never heard of that. And effects on the body are increased heart rate and blood pressure that may dissipate over time. Effects when used, mild to intense perceptual changes with high variability in effects based on strain, method of ingestion, and individual differences. And again, these are all psychologically addicting. Not physiologically, psychologically. So, going more into depth on the different categories, we have depressants, which are drugs that suppress the central nervous system activity. Usually, GABA agonists, which have a quieting effect on the brain, work by binding the GABA receptors, which makes the neuron less likely to fire. Again, that's what, and we went on that previous unit, unit 3. So these include alcohol, barbiturates, so anticonvulsant medication, and benzodiazepines, anti-anxiety medication. They also, alcohol is a depressant, so it decreases your reaction time and visual acuity, lowers levels of alertness, reduces behavioral control, and can result in complete loss of consciousness. And, if you guys are in college or, have seen some people or had it happen to yourself, you know that alcohol does cause all these. Like I've seen people pass out drunk, do really stupid things when they're drunk. So definitely not good to drink too much. But everything in moderation, right? All right, so what is GABA? So the GABA-gated chloride, or Ci- channel, is embedded in the cell membrane of certain neurons. The channel has multiple receptor sites where alcohol, barbiturates, and benzodiazepines bind to exert their effects. So this opens a chloride channel and allowing negatively charged chloride ions, Ci minus again, into the neuron cell body. So changing its charge in the negative direction pushes the neuron away from firing, thus activating a GABA neuron, has a quieting effect on the brain. So here we have this nice diagram. So, has multiple receptor sites right here for alcohol, benzodiazepines, barbiturates, and they bind to exert their effects. So it opens a chloride channel and it allows negatively charged chloride ions into the neuron cell body, which, is shown right here visually for you. Because visuals help out with that kind of thing. It's easier to visualize something sometimes than just think about it. So next we have stimulants, which increase overall levels of neural activity. So usually dopamine agonists, which work by preventing the reuptake of dopamine. Dopamine activity is associated with reward and craving, therefore these drugs can be highly addictive. These include cocaine, amphetamine, cathinins, so i.e. bath salts, and MDMA. Side effects can include nausea, elevated blood pressure, increased heart rate, feelings of anxiety, hallucinations, and paranoia. So here we have a nice picture of some crack rocks. So crack rocks like these are smoked to achieve a high. Smoking a drug allows it to enter the brain more rapidly, which can often enhance the user's experience. So, dopamine agonists. As one of their mechanisms of action, cocaine and amphetamines block the reuptake of dopamine from the synapse into the presynaptic cell. So you can see right here, reuptake is being blocked by the cocaine. This results in a larger amount of dopamine in the synapse right here. So that's what makes you feel so good. You have a larger amount of dopamine than you normally would. Next, we have nicotine and caffeine. Caffeine, I combined the two. Caffeine and nicotine are also stimulants. So caffeine involves antagonizing adenosine activity, and it increases levels of alertness and arousal. Now, nicotine interacts with the acetylcholine receptors. It is highly addictive, and it plays a role in arousal and reward mechanisms. Nice picture of coffee, in case you didn't know what that looked like. Next, we have opioids, which serve as analgesics, I think I said that right, which decrease pain through their effects on the endogenous opioid neurotransmitter system. They are highly addictive and include heroin, morphine, methadone, codeine. So some nice pictures of opioids there. And then we have hallucinogens, which cause changes in sensory and perceptual experiences and can involve vivid hallucinations, variable with regard to the specific neurotransmitter systems they affect. So mescaline and LSD are serotonin agonists, and PCP and ketamine are NMDA glutamate receptor antagonists. So, figure 4.21. So psychedelic images like these are often associated with hallucinogenic compounds. So if you're hallucinating, you might see something like this on hallucinogens. So medical marijuana shops are becoming more and more common in the United States as states start to legalize marijuana. So outside of drugs altering your state of consciousness, we have hypnosis. So hypnosis is an extreme focus on the self that involves suggestive changes of behavior and experience. Clinicians may use relaxation and suggestion in an attempt to alter the thoughts and perceptions of the patient. It has been used to draw out information believed to be buried in someone's memory. Unlike portrayals in the media, individuals undergoing hypnosis are in control of their own behaviors. So you're not going to see somebody like in a movie, somebody gets hypnotized and is forced by the hypnotist to do something. That's not how it really is. People vary in their ability to be hypnotized. So its uses include pain management, treatment of depression and anxiety, quitting smoking, and weight loss. So here we have popular portrayals of hypnosis have led to some widely held misconceptions. Again, like being able to hypnotize people to, like in this picture, play in a band or do whatever you want. That's not how hypnosis works in real life. Now, continuing with our other states of consciousness, we have meditation. So meditation is the act of focusing on a single target, such as breath or repeated sound, to increase awareness of the moment. Meditation involves relaxed yet focused awareness, and it shows promise in stress management, sleep quality, pain management, and treatment of mood and anxiety disorder. So figure A, we have this is a statue of a meditating Buddha, representing one of the many religious traditions of which meditation plays a part. And then figure B, people practicing meditation may experience an alternate state of consciousness. So these are two other types of other states of consciousness that don't involve ingesting drugs. So that was Unit 4. Sorry that I've stuttered a bit more. It's funny because we were talking about sleep deprivation. I've been a bit sleep deprived lately, so I'm trying to get that under control. Maybe I need to try meditation or hypnosis. But I hope you guys found that unit interesting and educational. Again, my name is Prescott. I'm here at PsychSync, Succinct Psychology, and that was the OpenStack Psychology 2E Chapter 4 lecture slides. So hope you guys enjoyed and have a great day, and I will see you in the next unit. Bye-bye.